All right, hi, welcome to Chapter 2 in HDFS 101 on genetics and prenatal development. So this is the one time in the class where we're skipping a full section of a chapter. So we are only covering sections 1 and sections 2. We're not covering section 3 because we go into more depth in that in HDFS 310. All right, here's our learning objectives for Chapter 2. We're going to be talking about the genotype, the phenotype, reaction range, genetic inheritance. Then you're going to be learning, making sure you can describe the sex chromosomes, how they affect us, what makes them different. And then we're going to be getting into some kind of more emerging topics around epigenetics and differential susceptibility, which are related to gene-environment interactions. Okay, let's get started with some basics about genetics. And if you are like a biology major, biomedical sciences, like this is so basic, you can probably just quickly skim it and know that you know everything. However, if you were like me and you weren't really paying attention to genetics when you learned it in 8th grade or in high school or in your Life 102 class, maybe like me, you were just thinking about what you were wearing and who you had a crush on, or you were sleeping. So maybe you don't know it yet, and so you need a little bit more of this information. Either way, it's here for you. Okay, so I really didn't find this interesting until I was having kids, and so hopefully you already have found it interesting. But if not, I would really encourage you to reread this chapter when you or your partner, or if you or a partner ever become pregnant. This will be more helpful than anything you can find in a baby book. All right, so humans have 46 chromosomes, or 23 pairs, that are in the nucleus of every single cell in our bodies. And these pairs of chromosomes kind of look like they're attached in a little X, but they're actually just very close together. And then the chromosomes contain that twisting double helix of DNA. And then if you kind of segment that out, those base pairs out, those become our genes. And we have 20 to 25,000 genes in every cell. And how genes take effect is they code for proteins. That's how they take action. Almost each person has 2 copies of each gene, one inherited from their biological father and one inherited from their biological mother. And each of these is called an allele. All right, let's take a look at these 23 pairs of chromosomes. So as you can see, except for the XY pair in males, all pairs of chromosomes are what are called homologous, meaning they are the same general length, and they also carry genes of the same type. So for females, all 23 pairs are homologous, but for males, the Y chromosome is a much smaller chromosome, and so the XY pair is termed non-homologous. Okay, so you may have heard of the Human Genome Project, which was an international project that began in 1990 and was completed 13 years later in 2003, that was mapping the entire genome. And I'm old enough that I actually had a friend from college who worked on this after we graduated. And you've probably heard this metaphor, but the Human Genome Project was like taking a book in which there's no spaces between the words and no punctuation and figuring out what were the words and sentences. So it was about kind of looking at the DNA within each chromosome and then figuring out where where one gene started and another gene, and where a gene stopped and where another one started. We were surprised in the Human Genome Project to find out that we only have about 25,000 genes. We expected, because we're so complex, we would have about 100,000 genes. We have about the same number of genes actually as mice or fruit flies. This is just an example of chromosome 9, which shows kind of all the genes that are on chromosome 9 that are related to different traits. Okay, so we share anywhere between 94 and 98.8% of our DNA or our genome with chimpanzees and something similar with gorillas. The reason for the range is that geneticists have come up with a variety of ways of kind of determining how similar and what counts as similarity. So there is some discrepancy there. But 99.9% of DNA is the same for all humans, which is pretty crazy. only 0.1% of our genome varies across humans. So then if we think about biological siblings, fraternal twins, and biological children and parents, they are sharing half of the genome that varies on average. So they are then 99.95% the same. And I want to just point out that fraternal twins and biological siblings have the same, they share the same amount of DNA. They're not, fraternal twins aren't more similar. Basically, fraternal twins are just like, genetically just like biological siblings, but they happen to be conceived at the very same time. Then you can see biological grandparents and their grandchildren share about 25% of the genome that varies. So then they are 99.925% the same. And identical twins have 100% of the same genome. And we're going to talk more about how they can vary epigenetically. All right, so let's talk about some key terms in terms of the genetic expression of traits: genotype, phenotype, and reaction range. So your genotype is all of your genes, not just the ones that get expressed through protein manufacture or turned on through epigenetics. And it can include both your dominant and your recessive genes. Your phenotype is what is actually showing up in your physical traits or in your behavior, which we call your behavioral phenotype, a term that's often used in relation to developmental disabilities. So when you're the carrier for something, it's showing up in your genotype, but not in your phenotype. So here's an example here in some pictures. You can see this is a picture of my husband and I when my son was born. And you can see that we both have dark hair, and yet my son has kind of red or auburn hair, which was a surprise to us and raised some eyebrows, I have to say. People were looking at me like maybe I'd been unfaithful. But what this means is that either Jordan or I or both of us had red hair in our genotype, even though that wasn't being expressed in our phenotype. And so we were carriers for red hair genetically, and we passed that along to Isaac, and then it was expressed in his phenotype. I was one time explaining genotype and phenotype to my daughter on the front porch. She was young, and after I explained them, she's like, "Oh, I get it." She's like, "I bet I have slow running in my phenotype because of you, but I don't have," oh no, sorry, she said, "I bet I have slow running in my genotype because of you, but it is not in my phenotype because I am a fast runner." So you might ponder, like, what's in your genotype that you don't think is showing up in your phenotype? And then lastly here, this important concept of reaction range. It's the range of possibilities associated with any genetic trait. So let's think of IQ. Maybe my genetic, I'm going to think mine is high, right? I'm going to hope for the best here. Let's think that my genetic reaction range for IQ is between the average 100 and 115. And so if I have a more enriched in-home environment, then my IQ tests are going to reveal a score of closer to 115. But if I grow up in a more impoverished environment, then my scores are going to be closer to 100. We can apply this reaction range to almost all of our traits, or many of our traits. So for example, music ability, right? We all have a reaction range for that could be on the low end, the middle end, or the high end, but where we land in our own range is going to depend on what kind of access we have to musical instruments and musical training. Our reaction range for skin cancer can vary, right? We all have a little range there, but it's going to depend on how much sun exposure we have. Similarly, our reaction range for athleticism. Some of us have got really the genetics for that. I don't, and so even with all, I could have trained a lot and I still was never going to be an Olympian because my genetic reaction range for athleticism was, average, maybe even below average. Okay, you can test yourself on this question. The totality of an individual's heritable information is referred to as his or her, and this is genotype. So this is definitely a term definition question, which we don't have too many of, but we do have some of those on the exam. So this is a picture of two identical twins who are in their early 50s, and it shows how that even though they have the same genotype, they now have different phenotypes. The twin on the left has a history of smoking nicotine and also has had more sun exposure, and so they look different than the twin of the same age and same genetics on the right. And so this shows us kind of genetic environmental interactions, and also kind of introduces us to this idea of epigenetics, that some genes can be turned on and some genes can be turned off, which we'll be talking more about. All right, so I bet this is familiar to you, but obviously our traits are determined by pairs of genes, one allele from our biological mother, one allele from our biological father. And as you may remember, you can be homozygous, where both alleles for a trait are the same, like the big B, two big B's or two little B's, or you can be heterozygous, where the alleles for a trait are different. And then, as you've learned, for many genes, there are some that are more dominant and some that are more recessive. And so then the dominant genes are both in the genotype and in the phenotype, but the recessive genes aren't expressed, are just a part of the genotype, not a part of the phenotype. And so in general, for a recessive gene to be expressed in the phenotype, it's going to have to be paired with another recessive gene. Okay, so when we look more specifically at heterozygous pairs, like the big B, little b, we often talk about the idea of complete dominance, where only the dominant allele is seen in the phenotype and the recessive allele isn't expressed at all. But actually, a lot of traits involve what's called incomplete dominance, where the alleles average in the phenotype. So you can get wavy hair from alleles for both curly and straight hair, or you can get medium skin color from having one allele for darker skin and one allele for lighter skin. And another key example of this is heterozygous sickle cell, which we're going to look at on the next slide. Okay, so let's look at the sickle cell trait, which is associated with sickle cell disease, as an example of incomplete dominance. Okay, so if you have two parents who are heterozygous for sickle cell, meaning that they have both a dominant and a recessive gene, if you could have a child that has two dominant genes, which means that they will not develop sickle cell disease. You can have a child that gets the two recessive genes, and then they will develop sickle cell disease. Or you can have a child that is heterozygous for sickle cell, like the parents, and this child will be resistant to malaria and won't develop full sickle cell disease. It's called sickle cell trait. But it is a form of incomplete dominance. It's a blend. So the child doesn't look just like the child with the two dominant alleles, the child in pink here in the diagram. They look unique. Unlike the child in pink with two dominant genes, who isn't going to get sickle cell disease, but is now more at risk for malaria, the child in the middle, the child in yellow, who has one of each alleles, isn't going to get complete sickle cell disease, but is going to be resistant to malaria. Something about the sickling of the cells, that it's called sickle cell anemia or disease because the cells take a half moon shape, and those can kind of clog the veins and lead to risk of heart failure. So the child in the middle isn't as likely to experience full sickle cell disease and cardiac arrest, is more resistant to malaria, but is under kind of some extreme kind of hot and kind of extreme exhaustion from physical exertion, can experience cardiac arrest. And there's been some limited research on African American athletes, like for example football players in the United States, who are having cardiac arrest, and this is believed to be the cause, that their cells will sickle, less likely, but will sickle a little bit during extreme conditions that then puts them at risk for cardiac arrest. So this is also an interesting example from an evolutionary perspective, because you would imagine that sickle cell disease isn't something you want to inherit. Natural selection would be trying to prevent that. However, because it is a protective, having one or both of the sickle cell genes is protective for malaria, and you're more likely to die from malaria in Africa than you are from sickle cell disease, it is still kind of in the gene pool through natural selection. But to sum it up, what we're really looking at here is that the being heterozygous for the sickle cell is an example of incomplete dominance, or a blend. Okay, so even though we learn about kind of the Mendelian traits where a single gene determines, an observable physical trait, like whether or not your earlobes are attached, most things are polygenic, and polygenic inheritance, meaning that there's multiple genes in play. And so here's an example of just the multiple genes involved for skin color. But this could apply to eye color, or your weight, or your risk of addiction and substance use issues, or your susceptibility to various diseases. So when we're trying to get at this nature-nurture question, how much do genes determine traits? Typically how researchers do this is through two types of studies, twin studies or adoption studies. And just by the way, that picture is from Twin Fest, which happens annually in Twinsburg, Ohio, where I don't know how many twins come together, identical twins come together every year. Okay, so a little bit more about twin studies. I always figure if you're an identical twin, you've probably been asked to be in a twin study. So just so we're kind of getting our distinctions clear, monozygotic twins are identical twins, and they share 100% of the same genes. Dizygotic twins, also called fraternal twins, share 50% of the same genes, just like any other siblings. And then the researchers are often looking at whether or not these twins were reared together or reared apart. And so you can compare, for example, monozygotic twins who are reared apart, comparing them to on different traits to dizygotic twins reared together, to help kind of tease out what is nature and what is nurture. So for example, monozygotic twins who are reared in separate households are more common than dizygotic twins who are reared together in the same household in a number of things. They are more common, their blood pressure has a higher concordance rate, their physical mannerisms or gestures, their brainwave patterns, their IQ or intelligence, their personality, their degree of happiness, and their risk for various disorders like anxiety, depression, schizophrenia, obesity, and autism. So this is kind of telling us that, again, environment and genetics play a part, but in some areas genetics has an edge. Similarly, with adoption studies, studying, for example, the risk of alcoholism and comparing the incidence of alcoholism, the concordance of alcoholism between birth parents and the children that they gave birth to, or between adoptive parents and the child that they adopted. And essentially, if your birth parents struggled with alcoholism, then you are on average 4.5 times more likely to develop alcoholism. But if your adoptive parents struggled, then you're 2.5 times likely. So this tells us, again, they both matter, right? But genetics is playing a little bit more powerful role in the case of substance use. Okay, I'm taking you back with this question to research methods in Chapter 1. Studies of twins raised apart provide a good example of what? And in this case, it's a natural experiment, right? Because it's naturally occurring groups. You can't randomly assign people to be identical twins or fraternal twins or not. Okay, so let's talk about a term called X-linked inheritance and why males are more susceptible to some disorders. So on average, males are at higher risk for infant mortality, learning disabilities, intellectual disabilities, and a number of other disorders. And the reason for this is because males are XY, and the Y chromosome only contains about 241 genes, and the X chromosome contains close to 3,000 genes. So they got their X chromosome from their mother and their Y chromosome from their father, their biological father. And so if they inherited on the X chromosome from their mother a recessive gene. Most genetic disorders code on the recessive genes. So if they inherited the recessive gene on the X chromosome, then they don't have anything on the Y to counteract it out. That gene is just missing. You have over 2,500 genes that don't have a partner or another allele, essentially. And I'll give you an example of that on the next slide. So just as a note, to account for these genetic challenges, humans naturally give birth to 105 males per 100 females because the male babies are more likely to die, either as infants or at some point in their lifetime. Okay, so this I think just helps to see this visually. There's the X chromosome with close to 3,000 genes and 153 million base pairs. And remember, all eggs from the biological mother are X, and the sperm can be either X or Y. So the biological father determines the sex of the baby. So the X chromosome, again, contains only about a tenth of the genes that the Y chromosome, contains only about the tenth of the genes that the X does. And so if you inherited a recessive gene on your X chromosome as a male related to color blindness, hemophilia, muscular dystrophy, again, learning disabilities, developmental disabilities, there's dozens of syndromes listed here on this X list, and very few of them are listed on the Y. So then if you inherit that, then you're, again, more likely to kind of manifest the disease. It will show up in your phenotype as a male. And we're going to see an example of that on the next slide. Okay, so you can see here that we have a biological mother that is the carrier of a disease. And if she were to have a daughter who would have gotten her X, maybe gotten the X with the carrier on it, but then another X from her father, her biological father, she may not develop the disease, right? Because it's usually recessive, and so the dominant X from her father is going to override it. But if we have a carrier mother and a normal biological father, and the child inherits the carrier gene, the recessive gene from the biological mother on his X, now he doesn't have anything to cancel that out on his Y, and he is going to be affected. All right, let's talk for a minute about a more difficult topic, selective abortion and son preference, also called daughter aversion, which has been historically common in parts of Asia like China, India, South Korea. And the reason for the preference for sons and maybe selectively aborting daughters is that sons carry on the family name. Typically, sons remain in the joint extended family household, sons and then the women that they marry, and they become the ones that financially care for the son's parents, right? So they're basically bringing money in. Daughters leave the joint household and go into their husband's joint household, and they require dowries. So sons basically bring more money, the family name, and so that's the reason for the son preference historically. Currently, sex ratios are improving after kind of a problematic time. The normal sex ratio is 105 males or boys born for each 100 girls. And this is due to the greater genetic vulnerability of boys that we talked about, X-linked inheritance. So let's look at a couple of examples. China, which really, the son preference was really exacerbated by China's one-child policy, which existed from 1979 to 2016, so about 35 years, where you were penalized if you had more than one child. So people were only, kind of allowed to have one child, and then they wanted it to be a boy. And so at the height of that, in 2004, there were 121 males born for every 100 female. Now, or closer to that now in 2021, that's now down to 108 for 100 females. The picture there shows India's sex ratio, and you can see that once abortion was legalized, and ultrasound to determine the sex became available, then the sex ratio became fairly imbalanced. And then with campaigns by UNICEF and the World Health Organization, now we're seeing the sex ratio become more balanced. It has actually been illegal since 1996 for doctors to reveal the sex of a fetus, but families can come to other countries like the United States and determine the sex of the baby, the fetus. Just as an aside, the imbalanced sex ratio is higher among some religious groups, or was higher in India than others. So Sikhs had the highest, followed by Hindus, Muslims, and Christian Indians maintained a normal sex ratio the whole time. So it depends upon groups. Okay, let's talk more about genes in the environment, and more specifically, we'll be focusing on epigenetics and differential susceptibility. Okay, I really encourage you to find the link in Canvas to watch this really engaging video on epigenetics that will help you kind of understand it as we begin to talk more about it. It's going to go over the basics. Okay, so test yourself on the answers to these questions based on what you learned in the film. Can environmental experiences actually change the genes we carry? The answer to that is no. But can environmental experiences activate or silence a gene? The answer to that is yes. We'll talk more about that. Can that activation or silencing be transmitted to future offspring? And again, the answer to that is yes. Some epigenetic tags are removed at the time of conception, but some do get transferred, and we're still in the process of figuring this out. For example, like one thing I've wondered about that I passed along was fear of water, of big bodies of water, because when I was 10, I witnessed an alligator attack. So I've always been scared, right, of the ocean and lakes, and especially in Florida. And I was really determined to not pass that along, for example, to my son. And so I was always trying to be really chill about the ocean when we visited and kind of modeling not being scared. And yet I did notice that he was really scared of the water when he was young and reluctant to go in. And I wondered, like, did I pass along, some epigenetic tags around kind of fear of creatures with sharp teeth in water? You remember we talked about the Human Genome Project, and now we have the Human Epigenome Project going on, where we're trying to map all the possible tags to turn things on or off, but this is a much more difficult project than the Human Genome Project. So just to recap what's important for you to know, and you should have heard all of this in the video. So epigenetics means over the gene, and epigenetics are primarily methyl groups, which is one carbon atom with 3 hydrogen atoms, and histones. Methyl groups can attach to a gene and can turn it on or off. Typically it's off, but my understanding is it depends on where it's placed. And then histones can kind of coil or uncoil the double helix of DNA, which can change how much a gene can express itself. And so what you heard in the film is that there are then bidirectional exchanges between genetics and the environment. Epigenetics is happening all the time. When we experience big changes, like we go through puberty, right? There are naturally occurring kind of epigenetic tags happening to kind of turn some things on, turn some things off, so that the body can change for puberty or for pregnancy. My limited understanding is, as we duplicate all of our cells through mitosis, and what makes a skin cell different from a neuron, I imagine that that's epigenetic tags, right? Turning some functions off, turning other functions on. So our bodies are doing this all the time, which are going to kind of affect what we're experiencing in the environment, if we're pregnant. But then obviously what we're eating, our stress level, the amount of trauma that we're experiencing, our lifestyle is all changing our epigenetic tags. And again, some of those we're still figuring out which ones can get passed along. So this is one of the earliest and more famous studies on epigenetics. Bygren was a Swedish nutrition researcher. And it actually took him years to get the study published because other scientists believed it was impossible. They believed experiences died with the individual and did not get passed along. But what Bygren found in this remote village in Sweden that in the 1800s that had periods of famine, average food, and feast, that it was those experiences were continuing to affect the ancestors of the people who lived through those famine, food, feast decades later. And so you can see some of the findings on this slide, which you don't need to know for the exam, just kind of have a sense of idea, or an idea of it. Like basically some grandchildren died earlier on average due to things like diabetes and heart disease. More specifically, this was grandsons of boys who experienced a feast season before they went through puberty, granddaughters of girls who lived through famine when they were in the womb, basically prenatally or at birth, and paternal granddaughters of girls who had a sharp change in food somewhere in childhood. And so even though this doesn't show a clear pattern of whether or not feast or famine or change puts people at risk, it shows us that the food available to earlier generations is still affecting our epigenetics today. And so with our current, obesity concerns in the United States, I wonder how much of it is genetic and epigenetic. And I wonder how having, on average, food much more readily available during the last, 50 years is affecting us and will continue to affect our generations to come. So here's an example of epigenetics during prenatal development, which we're going to be getting more into. And so what these researchers found is that domestic violence experienced during pregnancy was correlated with an epigenetic change in the glucocorticoid receptor, or the GR gene, of the child when they were ages 10 to 19. So this was a retrospective study. It wasn't correlated with the same epigenetic changes in the mother, even though she was the one who experienced the violence. It was only for the baby who was developing in utero at that time. And we know that increased methylation of the GR gene is associated with decreased GR expression. And GR, or glucocorticoid receptor, is a regulator of the HPA axis. And that's obviously the hypothalamic pituitary adrenal axis that results in cortisol or adrenaline. And it's associated with our responses to crisis or fear or stress. And it can also be correlated with mental health issues. And we know that the HPA axis can be hyperactive or hypoactive, like too, amped up or too tamped down. And either of those are both associated with mental health issues. And so what this is just showing us is that, the tuning of the HPA axis seems to be associated with early life events through epigenetics, kind of even when we are developing in utero. Okay, let's start talking about one of my favorite frameworks, differential susceptibility, which is a gene-environment interaction theory. I think you're going to really like this. I think it'll be something that you take from this class and teach to others. So differential susceptibility is also called the dandelion orchid hypothesis. It was developed by Belsky and also Boyce and Ellis all around the same time in two separate research laboratories. And it basically says that our biology, our genetics, our epigenetics, influences how susceptible or sensitive people are to environmental factors. And so dandelions basically seem to be kind of average no matter what's happening in their environment. And orchids seem to be more sensitive to their environment. They, just like real orchids, they do well under really great conditions, and then they do really poorly if they don't have, for example, the right humidity and the right sunlight. Whereas dandelions kind of look like dandelions no matter whether they're growing in, the middle of a sidewalk or in a beautiful garden. Okay, so this is one of the creators, Tom Boyce, of the differential susceptibility or orchid dandelion hypothesis. I'd encourage you to watch some of his TED Talk Very helpful. And you'll note that he says on average about 20% of the kids that he was studying would be classified as orchids. I don't actually know what the overall number is in more recent studies. So in this lecture and then in upcoming lectures, we're going to be looking at a lot of graphs that look basically like this, which shows that orchids really change their outcomes based on their environment. So if it's negative, they're doing the worst. If their environment is more positive, they're doing the best. Whereas dandelions change very little. It's often a non-significant change between a negative environment and a positive environment, and they're kind of mid-range. And so what this means is orchids, again, have high susceptibility to their environment, and dandelions have low susceptibility to their environment. So each of these categories on this slide have been used by researchers as indicators of differential susceptibility. And I'm going to be giving you more examples of these. So we can do research where we determine what people's genetics are, and we'll talk more about that, or what genes are turned on or turned off, their epigenetics. We can also look at their endophenotype. This is basically endophenotype means biomarkers, basically internal biological phenomenon that have to be measured or detected, like through a microscope or a blood test. It's not something you can just see with the naked eye. So this might be your cortisol or your blood pressure or your heart rate. And then your phenotype is what's obvious and external and visible. And in differential susceptibility research, this is usually around your temperament. And so how much negative emotion are you showing as a baby? How much distress? How easily are you distressed? What's your threshold for stimulation? How cautious are you when approaching novel situations? How easily frustrated are you? And basically, babies with more difficult temperaments would be considered, orchids. Okay, so these are some of the most common genes, if we just start with genes that are studied in differential susceptibility research related to human development and family studies. We've got sensitivity genes related to serotonin, the 5-HTTLPR gene, and that's been associated with more intense emotion, depression, anxiety, and attachment. Then we've got a number of dopamine genes, one of those being DRD4. That's associated with things like attention, motivation, reward. So like ADHD-like symptoms, and external behavior, and also substance use. And then we've got the MAOA gene, which is is an enzyme that affects codes for an enzyme that affects dopamine, serotonin, norepinephrine, and that has been associated with more research on aggression. Okay, so let's look at a genetics example. So this was a study done at UCLA, and it was, I think, of both kind of students and staff on campus. So it was ages 15 to 33. It was not like a clinical population or anything. And it surveyed people on the amount of life events, positive or negative, that they had experienced in the past 6 months. And then it also measured their level of neuroticism. Neuroticism is one of the big 5 personality traits, and it's associated with anxiety. And then they also did a blood test to determine which gene of the 5-HTTLPR gene they carried, the plasticity allele, which is often referred to as the short allele, or the long allele, which is the non-plasticity allele. So basically, one version of that gene makes you more sensitive to your environment. One version of that gene makes you less sensitive, the long version. And so what you can see here on this graph is that there's 3 lines. There's the homozygous short, so the kind of true orchids who had both the plasticity alleles for the short version of this gene. Then we've got the homozygous long, the bigger dotted line, the people who had both kind of the non-plasticity alleles of this gene. And then we have the people who are heterozygous, one of each. And you can see that the pattern kind of that is shown kind of confirms how we understand differential susceptibility. That orchids who had the most negative life events in the last 6 months showed the highest amount of neuroticism, but orchids who had the most positive life events in the last 6 months showed the absolute lowest amounts of neuroticism. Dandelions, that line, even though it looks like it's going up, it's actually just non-significant. They didn't show a change in their neuroticism, whether or not they had lots of negative life events or lots of positive life events. And those that were heterozygous had one short allele and one long allele, had a pattern that was somewhere in the middle. So let's look at another genetic study. And again, I want you to have the basic idea of these, but you don't have to memorize kind of the actual study findings. So this study, rather than just looking at one genetic allele, it was looking at several, and you can see those listed there, and it was determining whether or not people had 0 to 1 plasticity alleles, 2, 3, or 4 to 5. And so you can see the 4 different lines there. And then it was basically investigating how parenting quality from low to high influenced your level of self-control as an adolescent male. And what you can see found here is that for the true dandelions who had 0 to 1 plasticity alleles, it didn't matter for their later self-control as an adolescent, whether or not they had great parenting or poor parenting. And then the amount of plasticity alleles you had kind of had a cumulative effect on how much of an orchid you were. And so the true orchids, or the most orchid-like people in this study who had 4 to 5 plasticity alleles, had the most dramatic response, meaning that if their parenting quality was poor, they were the ones that were acting out the most as an adolescent male. And if their parenting quality was high, they were like the best. They showed the highest amounts of self-control. Okay, so this is an epigenetics research example of differential susceptibility, looking at the levels of methylate, or the methylation of the glucocorticoid receptor plasticity allele. And so these were all children who had this, the plasticity allele, but for some it was turned on and some it was turned off. And so for the ones in which it was turned off, it was methylated, meaning that gene could not have its effects to make the child more sensitive to their environment. And so for those children, the quality of their caregiver's parenting did not have an effect on their externalizing behaviors, how much they were acting out. But for the children in which it was not methylated, meaning the gene was able to be expressed, then they were looking like the true orchids. Basically, if the quality of caregiver parenting was high, then their externalizing behavior was low. And if the quality of caregiving behavior was poor, then their externalizing behaviors were high. And so this just tells us it's not just the genes that you carry that predict whether or not you're a dandelion or an orchid, but it's also your epigenome, whether or not those genes have been turned on or turned off. Okay, so we've done research studies on differential susceptibility related to genetics, epigenetics. Now we're going to look at temperament. So this was a study measuring kids' temperamental sensitivity. And they did this in which they basically put kids through a number of tasks, and then they had observers decide how typical the child was on what behavior they showed, kind of their phenotype in various conditions. And so it was how reflective are they, how cautious, how inhibited, how much do they shut down when they're stressed, how much can they pay attention when things are going well, how compliant, how anxious do they get when things are unpredictable, or how rigid do they get, how empathic can they be in positive environments, how fearful, how cooperative. So the kids were rated, and then the kids that had the highest amount of temperamental sensitivity, the top kind of 30%, were considered the orchids. And then they also measured basically maternal parenting difficulties. This is essentially high parenting difficulties would basically be a parent that was more harsh and critical. And low parenting difficulties would basically be a parent that was more empathic, warm, and sensitive. And they did this by observing how the mother responded through a variety of supporting their child through a variety of tasks, and also what their perceptions were about corporal punishment or spanking. And so what you can see here is that the children who were the orchids, the top 30% of most sensitive, temperamentally sensitive kids, showed big changes in their internalizing behaviors. This is basically how unhappy, sad, depressed, nervous, or high-strung or tense they were, depending upon their mother's kind of good parenting or poor parenting. And so if the mother was more harsh, then we had kids who were showing high amounts of these internalizing problems. And if their mothers were more supportive and sensitive, then these kids had the least amount of internalizing difficulties. Whereas dandelions, that relationship was not statistically significant and so unaffected by the quality of the parenting. Didn't matter to them. They were kind of in the middle, right, in terms of their internalizing symptoms, regardless of how well their mothers were doing. Okay, let's look at the example of an endophenotype differential susceptibility study. This was on low-income families in the United States, kids ages around from infancy to 3 years. And so what they did is they brought these kids in and put them through a series of tasks, and they measured their cortisol, I think like 9 times over a 75-minute period. And then they kind of basically determined, who were the high cortisol children versus low cortisol. High cortisol children released more cortisol at the moment of stressful events, and then had a harder time kind of bringing their cortisol back down. And so it basically is measuring your cortisol underneath this curve, essentially, is how they do it. So it's, okay. So, and then they measured parental depression through self-report and child internalizing behaviors, again, through the CBCL, the Child Behavior Checklist, in terms of how emotionally reactive, how anxious or depressed, how withdrawn, how much they complain about somatic things like my tummy hurts. And then also externalizing problems. How much do you struggle to pay attention? How aggressive are you? So questions might be like, how much are your child's feelings easily hurt? That might be an example of internalizing. And externalizing might be like, how much do they attack other people or break something that belongs to somebody else? And so essentially, high levels of cortisol is a key indicator of basically people being more like orchids, more reactive or sensitive to their environments. And so what we can see here is that if a caregiver had low amounts of depression, orchids and dandelions were about the same in terms of their internalizing behaviors. And so the orchids actually aren't doing a lot better, like we saw in some of the other graphs, and maybe this is because it's just an absence of a hard thing, not the presence of a really positive environment. So we can see that with low caregiver depression, orchids and dandelions are about the same. But then if caregivers are at higher levels of depression, this is where we see dandelions stay the same in terms of their internalizing symptoms, but orchids really increase. And so orchids are much more affected by their caregivers, and specifically their caregivers' levels of depression, than dandelions are. So this is continuing with the same study. It was part 2, and they did an intervention. So I want to just talk briefly about what does the dandelion orchid hypothesis suggest about who responds well to intervention. So this was the same cortisol study, dividing kids into being high cortisol kids, the orchids, or low cortisol kids, the dandelions. And then they put them all through an experimental design where they were either just assigned to be a control group or an intervention group that received what was called the kind intervention, which was 10 in-home visits that were a combination of videotaping caregiver-child interactions and then coaching them on how to be better parents. And it was all based on this serve and return model where you're trying to have these kind of back and forth positive reciprocal interactions with your child, in which you're supporting them, encouraging them in play, you're letting them lead in play, you're letting them switch activities if they want. And so you're naming and kind of narrating what they're doing in a positive way. So it's basically teaching parents how to be good parents with their kids. And so this graph is just showing the orchids, the high cortisol children, and whether or not they were in the control group, which did not get the intervention, or the kind group that did receive the intervention. And basically what you can see here is for high cortisol kids that didn't receive the intervention, like we saw before, they were more susceptible to their caregivers' level of depression. And if their caregiver had higher depression, then they had higher externalizing and also internalizing behaviors, even though the graph here just shows externalizing. Yet, if they received the find intervention, which is the solid line here in the graph, basically it blocked the negative effects of the caregiver's depression on their behavior. It became a non-significant relationship between caregiver's depression and externalizing symptoms. So it means basically the intervention stopped the negative effects of higher rates of caregiver depression. And yet for the low cortisol kids, the dandelions, the difference between the control group and the intervention group was non-significant. It basically did not change their behavior. The intervention or the lack thereof didn't have any effects on their behavior, regardless of the caregiver depression. So what does this tell us? It tells us that if the orchids are the ones who are most likely to benefit from an intervention. And so you might be creating this really cool program and hoping for some positive change, and some people show it and some people don't. And it may be that the people don't are just dandelions. They are not going to be affected by their environment very much anyway, which includes your intervention. All right, so what are the implications if you think you're an orchid or someone close to you is? And this kind of answers these questions like, what's wrong with me? In some ways, why can't I just be more chill? Why can't I be more relaxed? Why do things bother me so much? And so basically, it is like you're wired to experience the world in a more intense way, for better or for worse. So the bad news is that things are going to hit you hard. And you're probably going to have to ask for help. But the good news is if you can get help and support, you can really be amazing. I think it's probably the orchids that keep therapists in business, and we're the ones who benefit the most from therapy. I'm sure you've already been thinking about this as I've been talking. What am I, an orchid or a dandelion? And what about my siblings, if I have them, or my significant other? And so you can be thinking, like, which one are you? And the way I figure this out a little bit is thinking about how much does my well-being change depending upon what's happening in my life. And so I do think of myself more as an orchid because when I was younger, my well-being really correlated with my parents' well-being, and it fluctuated a lot. When my parents were less stressed, I was doing great. When my parents got more stressed, especially like after my brother was born, then they put more kind of pressure on me academically, things took a nosedive for me. But yet when I hit high school and my mom went back to school to become a marriage and family therapist, my well-being began to just dramatically increase. You can be kind of pondering this, thinking about the people you know. I have looked up some of my plasticity alleles on 23andMe, and sometimes I have them and sometimes I don't, but I can't always determine what's been methylated or not. Just funny story, I do think this is one of the things students take from this class, and one student told me, that they had actually been playing beer pong in their backyard, and they saw a dandelion, and they stopped the whole game so that they could explain the orchid dandelion hypothesis to their friends. So maybe that will be you too.